All right, just going to give you a very, very quick update on where we are with the metastatic bladder cancer. And for those of you who know and who don't know, May is the Bladder Cancer Awareness Month, so pretty good timing. So if you all remember, when we started thinking about the guideline-driven evidence generation, we got into a little bit of conversation and decided to come up with a different approach in terms of starting with a clinical guideline and decomposing the guideline into structured decision node and see how patients flow from one diagnosis, here metastatic bladder cancer, to getting a treatment, evaluating the eligibility criteria for each decision node. Once we have that, in the second step, we will be able to evaluate the feasibility or validity of addressing the many, many questions that can come up within the OHDSI network or within the research network. And based on the feasibility and having the evidence gap that basically comes from the guideline group, and I'm just going to, as a reminder, we're partnering with the European Association of Urology in this work. So based on the evidence gap that comes from them and also what we know from the feasibility, we can have targeted studies where we can do that and where high quality RWE can fill significant evidence gap as it comes to relevance, adherence, generalizability, and all that. So our key objective for this, the initial step, is to assess the relevance of guideline recommendation, adherence to guideline recommendation, and then the generalizability of what we see in the guideline. So if you think about it, this is a very, very simplified sort of like schematic representation of what happens to the metastatic bladder cancer patients. Some of them are eligible for treatment according to the guidelines, which fall into one of these four big treatment eligibility buckets. And if everything happens perfectly, they would get the treatment. So they would get a cisplatin-containing carboplatin, EV plus P, as you see, or those who are not eligible, they get the best supportive care. Reality is different. Some people get treatment, and some will get one of these four buckets, and some will get something else. We don't know what it is yet. A little bit looking deep, some of these treated are eligible according to the guideline, and within each of these components, they're eligible and they're ineligible patients. So if you look at these basically plots or figures, you see a lot of different cohorts, right? So a cohort of metastatic bladder cancer treated with different treatments, and eligible and treated, and so on and so forth. If we compare these cohorts, if we look and be able to look at the characteristics, we can assess relevance and adherence to treatment by looking at the outcomes of any of these, especially the treatment eligible who have received treatment, or in general, we can look and evaluate the generalizability of guideline recommendation in clinical practice. That's our general goal. Where are we today? So we've completed the first step. We've dissected everything from the guideline. Everything has been translated into concept sets, all available in Teams. We have reached out to our data partners. So far, we're very lucky and fortunate that 28 data partners have agreed to be a part of this. This is a distribution of where the data is coming from. Pretty global. I'm very excited to work with everyone. In terms of data partners engagement, we are working, 24 of our data partners have already done the oncology data readiness assessment, 4 are in progress. We have some additional quality metrics that we're gonna work on a lot, because a lot of these eligibility criteria are based on labs, and we know we have to work a little bit harder on harmonizing labs and units and measurements and coming with decision of what we can do and what not. So that's coming next. Study protocol is almost there. Will be distributed tomorrow along with everything else. And generally looking forward to continuing the work. If there are any other data partners that are out there, it's an open invitation. We would love to have you and make it even a bigger study. Thank you very much.
Great. Thank you so much, Asieh. Next up is Tatsiana.
I'm going to talk real quick today about our progress on FEP2 study, which stands for First Episode of Psychosis Pharmacotherapy. So we started in January with this idea of a head-to-head comparative effectiveness study for treatment of first episode psychosis, where we would compare all the antipsychotics recommended and mentioned in the clinical guidelines. Thankfully, there are not that many of them, in terms of frequency of rehospitalizations with a relapse that happens. Luckily, our general study idea did not suffer any major changes along the way. We just limited our number of antipsychotics a bit to only those that we can actually expect to see in the data in normal, big enough numbers. And so starting from January, we did our literature search. We created our clinical description as part of Phenotype February. We joined that to the initial study idea, and we created our phenotypes, which was a big, big chunk of work. We faced some challenges, and the phenotypes are quite complex since we're studying and trying to model basically the first episode of previously drug-naive psychosis and its relapse. And as you can see, we have like many inclusion-exclusion criteria and so on. Thanks to phenotyping working group and Anna specifically, we had several rounds of cohort diagnostics done, and that really, really helped us. And if you wish, the latest results can be found via this QR code, so you can look at them. And then in March, we compiled all the requirements we have to the databases, and thanks to Claire, we had data diagnostics run, and we have contacts with the data partners whose data fits our study model. And we have already reached out and contacted to them, and some of them have reached back, and we're planning to work together, and that's wonderful. Also, several of those data partners have already run their preliminary data readiness study, so we're good to go. And speaking of where we are now, we have our repository created, we are working in it. We have the first version of our protocol published, and it's been distributed to the working group, psychiatry working group, and other study members so that we can gather some feedback, and it will be sent out to the data partners real soon. So if you're our data partner and if you're listening to it, I'm gonna get it back to you real soon. We have the study specifications and corresponding script ready and basically in testing. And if you wish to look at any of that, that's open, so you can go through these QR codes and look at that. And so our next steps would be doing these finishing touches, responding to comments that our working group might have in the protocol and scripts. We're going to once again get back to the data partners and give them the protocol, the specifications, so that they could run the study. We'll collect results, assess them, hopefully publish. And so I wanted to once again say huge thanks to all the working groups and individuals who helped us get to where we are. Thanks to Claire, thanks to Anna, thanks to phenotyping and psychiatry groups. And if you're interested to join, there's still time, definitely. Feel free to reach out to me via any of these channels. And thanks for listening.
Great. Thank you so much, Tatsiana. Next is Chen Yanover.
So we are looking into use of osteoporosis medications in individuals 75 years old or more. And we know that just a little bit of background, this group is at the highest risk of fragility fractures. But when you look at RCTs, you see that in most cases they are poorly represented or even ineligible to participate. And when you look into observational studies, in many cases, the numbers of this subpopulation is relatively small. And in many cases, they are not analyzed as a specific subgroup. So we look into gaps in the guidelines with regards to this subpopulation. And specifically, our study addresses some of these challenges. So we first want to understand the patterns of using osteoporotic medications, and then we want to compare the effectiveness and safety of different drugs within classes of drugs used to treat osteoporosis. And a slightly more complicated study design that we look into is look into the safety and efficacy of long-term use of some of these drugs, and we talking about 10 or more years of usage. So where are we? So we implemented most of the phenotypes during Phenotype February, and, with the help of the phenotype work group, and used the core diagnostics data to fine-tune these definitions and improve them. We did that for the indication, for the exposures and outcomes, and for many of the covariates. Then we looked at data diagnostics and identified databases that may be relevant for the study, and reached out to data partners and heard back from quite a few. And we also have the first draft of the study protocol just completed, and it's available in this link and also in the study Teams channel. And we'll be happy to get comments on this protocol. And next, we plan to implement the analysis and understand how to get relevant statistics from the treatment patterns package. And for these last two tasks, we'll again be happy to get help from relevant people in the community. As collaborators, beside the initial collaborators, we also been joined by a collaborator from Australia, and we'll be again very happy to see more of you if you're interested in joining this study. Thank you.
Great, thank you, Chen. Next up is Chris Mecoli.
So, hello, everyone. Thank you for providing a forum to give an update. My name is Chris Mecoli. I'm one of the investigators at our Johns Hopkins site here in Baltimore in the US, joined by our team, Ben Martin, Will Kelly, Christelle Zhang, and Eric Westland. So I'm going to give a quick update on the rheumatic disease treatment and infection risk study that we started several months back. All right, so recap for why we're doing this. Infection is a major cause of morbidity and mortality in rheumatic diseases. It's always in the top 3. In many rheumatic diseases, it's the #1 cause of morbidity and mortality. And little known, despite that, is known about the less common rheumatic diseases and infection risk for various medications that we use to suppress the immune system. Most of the work here is done in quote unquote more common rheumatic diseases such as rheumatoid arthritis, but some of the less common ones such as lupus, scleroderma, myositis, Sjogren's, there's really not a lot of data here. And there's really almost nothing known about combination therapy. So the data that exists is on individual medications, but that's not how we practice in real life. The majority of patients receive combinations of immune suppressive drugs, and we don't really understand the risk of these combinations. This is reflected in all the evidence-based guidelines that exist. You hear and read this theme of need for additional studies addressing these issues come up again and again. So that's what prompted us to do this. So what's been done so far? We created our target phenotypes and cohorts informed by the literature. So you can see some of the autoimmune diseases of interest: lupus, myositis, scleroderma, uveitis. We also looked at all the drug exposures that we tend to use, and then we developed our infectious outcomes, namely shingles, pneumocystis pneumonia, and PML. We iterated these phenotypes via the cohort diagnostics runs, multiple runs by OHDSI. So thanks to Anna and her team for facilitating that. We also modified based on expert input from other rheumatologists and infectious disease specialists that are in the pharmacoepi space. Then, thanks to Claire and her team, we did a feasibility check across the OHDSI Evidence Network, which was awesome. Really exciting to see how many data partners there are and that have the ability to actually run at least some of our analyses. So that was great. What we're working on now is really sinking our teeth into the study design and protocol. We realized this is my fault. I probably bit off a little more than I can chew. I got overly optimistic. We eventually had ideas of looking at both infection and cancer as outcomes that proved to be overly complex. So we basically simplified our view of just going after infection for at least this first network study. We are continuing to solicit feedback from other pharmacoepidemiologists, and we're trying to take into account the fact that we're leveraging both EHR and claims data as we develop this study design, because there are obviously differences and pros and cons of each data source. We're also in parallel working on creation of a strategist test package and working on contacting and confirming data partners. We've heard back from several already, which is also wonderful. So what we could use help with, really, I'm looking for people to tear apart our study design. So hopefully in the next month or so, we're going to publish on Teams a first draft of the protocol. And I'd love people to weigh in on the rigor of our design, our sensitivity analyses that we are proposing. Are there any red flags based on the expertise in the room that we can avoid pitfalls that others might have had in the past? We'd also like some help reviewing our strategist test package code, which is in the process. And then, of course, any other data partners that are interested in participating, acknowledging the fact that you probably want to see a protocol before you sign on to this study. Feel free to reach me at that email address, and I'll be happy to either set up a call or we can just communicate via email moving forward. Thank you for the time. Oh, and then timeline-wise is to have this all kind of ready to go at least in the mid to late summer, if not earlier. So thank you.
Great. Thank you so much, Chris. I will have slides posted afterwards, so I have a number of the slides, but anybody else, please send them to me after this call. Okay, next up, we have Oleg Zhuk.
Yeah, thanks, Craig. I don't have slides, and actually now I think that I should have prepared something to show you. So I represent the AKI study, and the idea study is very simple. We basically want to answer the question, how many patients after certain surgeries develop an acute kidney injury, even though it seems that this question has been answered many, many times, but the data is very, very different from study to study. We have some studies showing only 1% of AKI after major surgeries, but that's sounds like a nonsense. And some studies show more than 30% of acute kidney injury, even though they should be in at least in one range, in one diapason. And this is the idea, but since then the study grew actually bigger than expected. So we focus on AKI as we planned originally, but we also decided to include more surgeries and more surgeries and more outcomes, also surgically related, such as sepsis or clostridial colitis, that are very, very relevant in these surgical populations. Also, I want to, I'm very proud to say that we recruited at least one OHDSI newcomer, and this is already a great result of our study. And I want to thank everyone in the community for the engagement and participation. We had a fantastic February, very productive. And shout out to the Phenotype February team. We changed lots of our phenotypes based on the iterative runs of cohort diagnostics, many of them. In March, thanks Claire and the team, we identified potentially relevant databases for our use case, but we haven't contacted the data owners yet. And as of now, we're finalizing the protocol. The draft is available on our channel. We also created a GitHub repository, but it's still kind of in a draft mode. So next steps for us are basically very simple. We need to finalize the protocol, and it will be done soon. And the idea is to contact data owners with the protocol and when it's ready. This is it. Thank you very much.
Chungsoo, do you want to go, and then we'll try to get back?
Okay. Yep. So yeah, hi everyone. I'd like to briefly reintroduce our project on the cardiovascular events in oral and new anti-obesity medications. And this is part of the 2025 OHDSI Guideline-Driven evidence generation project. I'm sharing this to update the community and also to refocus our effort on this important work. So back in January, we identified several evidence gaps on anti-obesity medications in clinical diabetes and obesity guidelines, particularly the lack of head-to-head comparison, long-term follow-up studies, and evaluation of effectiveness beyond the weight loss, such as cardiovascular outcomes. So to address these gaps, we launched a comparative effectiveness study across the OHDSI network, focusing on cardiovascular outcomes associated with new and old anti-obesity medications. Just 2 weeks ago, I attended a webinar hosted by the U.S. National Institute of Diabetes and Digestive and Kidney Disease on GLP-1RA and real-world evidence. The discussion highlighted key evidence gaps, particularly around long-term outcomes, persistence, adherence, and comparative effectiveness study, which closely aligned with gaps we had previously identified. And also several presenters emphasized that the importance of using distributed research network to conduct studies across multiple databases and countries. So, and also OHDSI was mentioned multiple times as a key example of such a collaborative framework. To summarize the research background, semaglutide and tirzepatide have recently drawn significant attention due to their cardiovascular protective effects. However, there remains limited evidence comparing their cardiovascular outcomes with those of earlier chemical-based anti-obesity medications. So based on the hypothesis that these novel agents may offer superior cardiovascular benefits, we aim to conduct a comparative effectiveness study. Let me share a brief update on our current progress. The study protocol is currently under development, while the introduction section was relatively well defined, but the method section is still being drafted and includes several areas that require further discussion, such as how to handle different definition of obesity and how to define the study population, including whether or not to include patients with diabetes. And there are some concerns about the compatibility due to the obviously varying weight loss efficacy across medications and other additional outcomes as well. So we are very open to community, your input. So if you're interested in the collaborating, please feel free to leave any comments on this study channel. And I'd like to special thanks to Jack, Samir, and Shanshan for their active engagement for this project. We've completed the cohort diagnostics for the initial for checking the initial study population with Anna's support. The diagnostics were run on the CCAE databases, and with Cindy's from Colombia, her help, I was able to identify some significant several errors. So as a result, we were able to see some decent number of patients, especially thanks to Anna and Cindy. Next, I'll be focusing on refining the protocol, and my goal is to have a solid draft ready within next month. And there's still a lot to sort out, so I really appreciate everyone's input and support during this phase. And once the protocol is finalized, we will share it with potential data partners to begin some kind of administrative steps like IRB submissions. And in the meantime, we will get started on building the study packages. that's an area I'm quite comfortable with, so I hope to keep things moving quickly. And I'm aiming to complete at least one round of this analysis by September. That feels like a realistic but exciting milestone. So yeah, that was my presentation. I felt our study was falling behind due to other competing priorities, but I wanted to bring it back and refocus our efforts. Bad news is time flies, but good news is we're once piloting this study and can steer it forward. Thank you for listening.
Great. Thank you, Chungsoo, and thank you to everybody for your leadership in these studies and continuing to push them across. Okay, we have Chang Hoon back, so let's try again.
Thank you. I'll proceed like this. I don't know what went wrong last time. Sorry about that. So what I was saying was that our study was focused on comparing these two drugs, ticagrelor and prasugrel. If we briefly cover the background again, P2Y12 inhibitors were used in acute coronary syndrome undergoing percutaneous coronary intervention. Comparing these ticagrelor and prasugrel, ESA React 5 was the RCT that compared these two drugs. But due to its limitations, the superiority of these two drugs is not yet fully established. And two major guidelines, European and American guidelines, have contradicting recommendations. So our goal was to address this evidence gap in the guideline, and we followed the overall timeline of the GD 2025 study. I believe this was the overall schedule we were set on. So in Phenotype Library, we built the cohorts, and because this study mainly focused on outcomes, cardiovascular outcomes, luckily I could get many of the phenotypes and cohorts from the well-established LEGEND study. However, after reviewing the phenotypes, there were some modifications made to the cohort definitions and phenotypes to better fit the clinical descriptions of my study. And thanks to Anna, I could run the core diagnostics on multiple databases. And thanks to Claire, I was able to access the DB diagnostics results. So these were the lists I got from the DB diagnostics, and we contacted them to if they can engage in our study. So after that, in April, we wrote the first study protocol, and the first version of the study protocol is already published in HME catalogs. And we also built our first version of our study package, and it was uploaded to a public GitHub repository. So we are still in this step of the overall timeline because our protocol and the study package still requires some more revision and some more discussion. So I won't go too deep into the current protocol, but this is the overall design of our study. We are going to compare effectiveness and safety of prasugrel and ticagrelor, and the cohorts and study design was designed this way. We also set the diagnostics thresholds and major requirements needed in the protocols. And what we did after was to request a review from a clinician, and we learned from this that our current protocol is most of the people who are already familiar with the CDM and concept IDs and those concepts can easily understand what we wrote in the protocol, but we found out that it was very unfamiliar language to the clinicians. So such as usage of term of on treatment or intention to treat, well, this originates from mimicking the RCTs, but since this is the observational study, it was pointed out that we should consider using as treated or initiator analysis. For the definition based on concept IDs, these were not as clinically understandable to the doctors. And
I think we
Yeah.
lost him once again. Just let me briefly end this. So there is no further slide beyond here. So currently we are trying to finalize the protocol and package based on the comments we've got, and then we are trying to run this package against the nationwide Korean database, which is HERA. And I hope that the analysis of individual centers and meta-analysis can be done by June. Thanks much.
Great. Thank you, Chang, Hoon, and to Chan for your teamwork there. Next up will be Anna.
This is an update on the community-acquired pneumonia treatment pathway study. And the goal for this study was to characterize antibiotic treatment of patients who had community-acquired pneumonia at scale. And the rationale for this study is that there are specific protocols on how to treat pneumonia as a first-line therapy, as a second-line therapy, but it seems that based on the anecdotal studies, clinicians do not really follow those guidelines, and this is where OHDSI could really help in assessing what are the real-world patterns of use of antibiotics in different populations and in large sample sizes. So with this idea in mind, we followed the study steps that you've already seen in multiple slides. We created the cohort definitions for patients with pneumonia as well as for drug classes. We assessed them during phenotype February. You have the link to the results here if you want to explore them. Then we moved on to developing the study protocol and specification. And because treatment pathways is a new module in Strategos, shout out to Sana who incorporated that into Strategos, we now need to also test it and see how it works on our local data prior to distributing the package to our network. So this is where we are right now in this middle box. And while we are testing the package, what I can learn so far, or module, what I can share the learning so far is that it does run on a smaller databases. And right now we're exploring the results of it to see what kind of visualizations we can use from it. Nonetheless, we face the challenge of a slightly worse performance on a larger databases. So this is where we spend the majority of our time right now trying to figure out how to proceed with the larger databases and with the current treatment patterns module. But as it relates to where the community can get engaged, the obvious space is to review the current protocol that we posted in Teams. And because it would be very nice for everybody who's interested to engage at this point and make sure that their comments are accounted for, that they're fine with the design, that they like what they see, I wanted to spend a couple of minutes on talking about the design itself. So what we are thinking of right now is that we want to take patients with incident pneumonia, where the diagnosis was associated with the outpatient visit. We're looking at community-acquired pneumonia, so we're excluding patients with tuberculosis a year prior, and we also exclude patients with other pneumonias that are not community-acquired on the day zero to make sure that we're not capturing those. And we also require them a year of prior observation. And then, aside from it, we want to examine the subgroup, which is patients who come in with community-acquired pneumonia and then are subsequently hospitalized, because we hypothesize that the treatment patterns in that subgroup will be different, and we want to be able to showcase it. Then we start following those patients when they receive a diagnosis, and we're looking for drugs, and we identify broad classes of drugs. And you can see on your right side, the drug classes. You can see that it's not only antibiotics, but also corticosteroids. And then for antibiotics, so far we are thinking just the classes, but then some of the classes are stratified by either generation that is relevant for cephalosporins, or by route of administration, because there is a difference between drugs being administered through IV versus tablets. And we want to capture that. But we're keeping it at the drug class level. So for example, if you're interested in participating in the study, you could go into the protocol, review the drug classes, and then maybe make notes of special antibiotics that you would like to see on their own, potentially. And then in terms of parameters, we follow those patients to see all of their drug exposures, starting with the diagnosis with the day 0. And then we're saying that if the drugs were administered within 30 days, that they would be collapsed into one single error. They would be treated as an exposure. This is, for example, and also an opportunity for you to chime in and comment on those collapse sizes, because, for example, the error collapse may be different for different antibiotics. So this is an opportunity for you to chime in with your comments. And we also say that because these are acute events and drugs are administered rather quickly, we consider a combination to be the drugs that are administered within 1 day, or at the most 1 day apart. And then if they're administered, for example, 2 days apart, we say that it's no longer a combination, but a subsequent treatment. And these are the types of parameters that are described in the protocol. And this, I think, is exactly an opportunity for the community to come in and comment and maybe add additional things that they would like to look at to enrich the study. So if you want to get engaged, you would go to the channel under OHDSI that is called Community Acquired Pneumonia Management, and you will see that there is a study protocol there, and please feel free to leave comments, additions, or shoot me an email if you would like to discuss that in more detail. Thank you very much.
Thank you, Anna. Next is Cindy Kai.
So as a quick recap for why we embarked on this network study, the clinical guidelines are very clear that patients with diabetes should be receiving at least annual diabetic retinopathy eye exams, and particularly for patients with type 2 diabetes, that first eye exam should be happening at the time of the diagnosis. And despite this very clear guidelines, less than 50% of patients with type 2 diabetes are receiving the recommended diabetic retinopathy screening, which is leading to diabetic retinopathy still being a leading cause of vision loss. And that's because we're missing early opportunities to institute vision salvaging therapies. And one of the fundamental problems is that there aren't enough ophthalmologists to do all of these diabetic retinopathy exams for our type 2 diabetic patients who need them. But thankfully, with the advent of technology, going to see your ophthalmologist or optometrist in the office is no longer the only way to receive a diabetic retinopathy screening exam. We also have telemedicine, where a fundus photo can be taken and that photo can be read remotely by an ophthalmologist, or autonomous AI, where a fundus photo is taken and the AI algorithm tells you whether or not that patient has referable diabetic retinopathy. But there are still gaps in evidence, mostly questions like how is autonomous AI diabetic retinopathy screening being used? And with all the advent of all this technology, are we doing a better job with diabetic retinopathy screening? So the whole purpose of our current network study is to characterize the current landscape of diabetic retinopathy screening, answer questions like among patients with newly diagnosed type 2 diabetes, what is the choice? How are they getting their initial screening? And is technology reducing the time from diagnosis to that initial diabetic retinopathy screening? So to illustrate where we are in the network study execution, I'm going to use this graphic designed by Ben Martin that lists all the different steps that we've heard about from the other presentations today. So we've done our base camp preparation. We've identified the exact scientific question that we want to answer. We've developed the protocol, and you can find a copy of that at our study GitHub website, which is listed below. Claire has thankfully, run data diagnostics, and she's identified the data partners within the OHDSI Evidence Network who have data that can be used to answer our study question. And we've worked with Asieh and the folks in the phenotype development team to develop our phenotypes. For example, our indication cohort of newly diagnosed patients with type 2 diabetes and our outcome cohorts of the 3 various types of diabetic retinopathy screening. And the core team at Hopkins, which includes Eric, Westland, Ben Martin, and Hannah, have been working with the help of the broader Odyssey community to finalize our Strategist pipeline, which calls on characterization and cohort incidents, as well as the new treatment patterns package. And so back to our study question, are we doing better with diabetic retinopathy screening with the advent of all of this new technology? And if you are as curious as I am to answer this study question, please help us push our network study up this execution mountain. So if you are a data partner in the Odyssey Evidence Network, you've already heard from me. I'm hoping to get you the study package, at least Strategist, by the end of this week. And if you are a data partner not in the Odyssey Evidence Network, but you know for a fact that autonomous AI is being deployed at your institution for diabetic retinopathy screening, please reach out. If you have experience producing study packages, we'd love to get help finalizing Strategists and treatment patterns as well. And of course, if you're a clinician, you can start exploring initial results at this website here and start generating insights for our eventual paper. And so if you haven't done so already, please subscribe to our team's channel to receive updates, and this will be a great way to get in touch with us. Thank you.
Great, thank you, Cindy. Last but not least is Gayathri.
All right, so I'll try to keep it very brief since I'm the last one on the line. So our network study is with regards to pediatric vision screenings. Just a quick overview of why we do vision screenings in young children. So vision problems can impact not only visual functions in the eye, but especially in early childhood, it can also have an impact on overall development as well as learning. That's why vision screenings are conducted periodically to detect these vision problems so that treatments can be provided at the right time. Current guidelines for vision screenings recommend that annual screenings be performed in children under 5 years of age and then biannually afterwards. And even within children, even between children birth to 5 years, if you look at the methods of vision screenings, instruments are recommended for vision screenings in children under 3 years who are nonverbal mostly. And then visual acuity charts using symbols or letters are used for vision screenings in children 3 years and above. There is some current evidence of benefit for vision screening in children 3 to 5 years of age, given all the studies that came out of the Pediatric network looking at early detection of amblyopia and the different treatment strategies that are currently available. But we have very little evidence on how vision screenings are done, with what techniques they are being done, as well as the benefit for vision screening in children under 3 years of age. So our study is primarily focusing on looking at this evidence gap in children under 3 years of age, but we are also hoping to characterize current practices for vision screening in children between birth to 18 years of age, the typical pediatric population. So since 2016, there has been an increased adoption of instruments to screen for vision problems in young children. So our hypothesis is that that there is more occurrence of instrument-based vision screenings and at increased levels across different regions, but we do not have clear evidence to support this claim. Excuse me. So far, what we have done with assistance from Claire is we have run data analytics to look at data partners, and we have up to 31 identified data partners across the world. But we still are going through phenotype development, particularly for vision screening results and how they are documented in electronic health records, as well as looking at the different eye exam components so that we can really look at the question of the benefit of vision screening in children under 3 years of age. So we look forward to continued engagement with the Odyssey group and get help in developing these phenotypes.
Great. Thank you so much. Thank you to everybody, all of our study leads for your presentations. Again, if you can share your slides with me so I can post to the community calls page. We'll also, of course, post these recordings. Patrick, do you want to do a quick kind of overview of where we are and maybe where we're going in the next month?
Yeah, thank you, Craig, and I'll echo the thank you to all of our study leads for giving an update. I think one of the interesting things that we're experiencing as a community this year is trying to simultaneously drive a whole lot of very different studies forward. And I think we've seen some really nice progress. Appreciate the leadership of all of our study leads. Heard a lot of call outs there for Anna for all the great work that she did during Phenotype February in trying to drive these forward, and to Claire for her efforts in the data diagnostics. I think one common theme I heard through a lot of these studies was we're still in the midst of trying to align on our study protocols to make sure that we're both clear about what we are trying to do and that we can represent those ideas consistently so that we can apply standardized analytics to generate the evidence for these studies. So I encourage everybody in the community to be engaged in these studies, for the study leads to make sure that we're using our channels that we've created in the Odyssey community so that everybody in the community can contribute, participate, share input, feedback. And then ultimately these protocols are going to be the basis for our data partners to be able to get approvals to execute the studies and share results. But I'm encouraged to see the progress that we're making all these studies. I think this also kind of connects into our plans for June. Craig mentioned briefly that we're going to really focus here on Atlas as a user interface tool that we use in the community for a variety of different efforts. Chris Knoll and Anthony Senna and the Data for Life team and various members of our Atlas work group are going to be spending some time sharing the progress and journey that we've taken from Atlas, where we're going, and where we need to be going together as a community. But I think that the core theme that connects all of these pieces together is that we recognize that if as a community we're going to get to the point where we can efficiently and reliably design and execute studies, that actually starts with making sure that we have clear and unambiguous specifications for our analyses. Those specifications very often start with making sure that we have very clear cohort definitions, and Atlas is one of our primary tools to facilitate the design of cohorts. But also that we need to look ahead towards how we can also improve the overall analysis specification process. And we've heard from Anthony Senna about how we're using Strategos, but I think it's also still clear, even from the presentations today, we don't yet as a community have a consistent, standardized, reproducible process that we're following for designing protocols, for communicating the ideas of our studies in a consistent way. And I think that one area for us to collaborate on as a community is to think about that process of study specification and think about how we can actually standardize that, inclusive of using our open source tools to help facilitate that process. And I think Atlas can be one of those tools that can support that. So I encourage all of you, as we get into thinking about using Atlas, I want you to be thinking about these guideline-driven evidence as kind of a motivation for how we can use open source tools as a way to help us collaborate together in the design and specification of studies, in addition to allowing our software to be something that we can facilitate the execution of network studies and the synthesis of results to bring things together. And so I think it's very helpful that we're going to be able to spend a few weeks together really diving into thinking about that problem at a meta level. And that'll also provide us, our study leads, the opportunity to get these protocols done, get the communications continuing to roll on in the Odyssey Teams channel. And then hopefully we transition these studies from design into execution and shared interpretation of those results. So thank you all again for all the study leads, and encourage all of you to be engaged in any and all of these studies as we continue forward, and to also think about what lessons we can take from these studies and how we can carry them forward into our own methodologic research and open source development to make sure that as a community, we can make this process as efficient and reliable as we can.